So we're talking about how we go from molecular shape to smell, but I'm going to start with an anecdote from my undergraduate days, but going from smell to shape. So I was a chemistry major as an undergraduate, and we had a identification laboratory where we got a vial of an unknown compound, and you have to spend the rest of the week putting it through NMR and IR spectra and mass spec and anything else you can do, all sorts of little chemical tests to identify what the compound is. And usually that takes 10 or 15 hours of your time. I had a friend who opened up his vial and he sniffed and he said, mothballs, I think that's naphthalene. And he closed it up and put it in the drawer and went home and played video games, and he was right. And he saved himself a lot of time. So molecular features are nice, but sometimes just smelling it, it's amazing how much we can do. I'm going to use this sort of color scheme of blue and red throughout just to distinguish between the things we have to predict with and the things that we want to predict. So I'm going to kind of blow through the first several slides because you've heard about it possibly 3 times already this morning, what was being done. We have this data with a large number of descriptors. These descriptors are kind of weird in the Dragon data set. So some of them are very normal and you think would be important for predicting things. Others are just have weird names, and I was a chemistry major. I have no idea what these things are. But some of them are used in the prediction, so maybe they're important for olfaction for reasons we don't understand. And what we're given is information about perceptual data for about 400 different molecules and all these subjects. There was obviously 2 concentrations, as you heard. And in some cases, there was a couple of extra replicates at the same molecule, at the same concentration, in the same subject. And we have to predict these descriptors for the holdout set, which is about 1/5 of the total data set. So, what these perceptual descriptors are. And there was, I'm going to expand upon the range of the descriptors. So there was actually 21 total: intensity, pleasantness. The other 19 were bundled into sort of one category for scoring purposes, but they're still each in their own right important. And so the only sort of missing data in the perceptual data was if a subject could not smell it, they rated intensity as 0, the other descriptors were skipped, which actually in some cases presents some interesting challenges in prediction, which I'm not going to go into, but you can ask me about later. So what I do to sort of prepare the data to be ready for use, essentially just make a matrix. Some of the descriptors of all these Dragon descriptors were very, very sparse. Some of them had all zeros because none of the molecules that were in the study were exotic enough to have non-zero values for some of these very strange descriptors. There was some missing data. If there was too much missing data, more than about 75% of the molecules had a 0 value, they were just simply removed. Otherwise, I performed median imputation, as opposed to some of the more sophisticated imputation techniques you heard about yesterday, to impute missing values so I could proceed with my algorithms. And I removed columns that were 0 variance. So if all the descriptors had a value of, Dragon descriptors had a value of 1, then it doesn't really convey any information about the molecule. So you're left with about 3,000 descriptors that vary across the molecules sufficiently to do some sort of estimation. I end up doing a random forest like Yunfeng, so the sort of normalization data here, which would be more important for something like a regression analysis or other algorithms, just to sort of get the data to be more normal looking. And then because there was 2 concentrations, I just duplicated each molecule and instead of Dragon descriptors, so there was 2 rows instead of 1 row for each column, and 1 row corresponded to the descriptor. Each row had all the descriptors, and then as the last column had the concentration as well. So I'll call this matrix X. And for the data, the perceptual data, there was taking the mean across replicates, because this is trying to predict the mean and the standard deviation. You get this sort of 3-dimensional matrix of molecules by descriptors by subjects. And depending on the goal, whether it's trying to predict intensity or one of the other things, I used either the high or the low concentration. Median imputation across subjects, so some of the subjects had missing values, right? And so I had to impute what would they have smelled had they been able to smell it. Which seems like kind of a strange thing to do, because why not just put 0? But it turns out that this actually works better. So we'll call that matrix Y, and of course we're trying to use X to predict Y. So the model fitting, I did random forest regression. Yunfeng just showed you what goes on. I did this in Python, the scikit-learn package. for some of the most complex parameters that required the use of the most features, they used a variant of random forest called extra trees regression to predict some of the parameters, more or less just intensity, which is by far the most complex of all the parameters. Excuse me, of all the descriptors. I grew one random forest for each descriptor. What that means is these decision trees, I grew about several hundred of the trees for each descriptor predicted independently, and then I moved on to the next descriptor. And I did just cross-validation with like 4-to-1 splits for to optimize all my random forest parameters, like how many features do they have, how deep can the trees go, how many samples do you need at each node of the tree in order to have that node. And so now the more interesting parts of the talk, some of the perceptual descriptors were very complex, meaning they required a lot of features from the Dragon data set, a lot of physiochemical features to predict accurately, or to predict as accurately as possible. Intensity was the most extreme of these. So what you can see is here's the 21 perceptual features arranged in a sort of 7x3 matrix, intensity, pleasantness, and so on. And on the x-axis is the number of features on a log scale. On the y-axis is the correlation between the prediction and then the actual values in cross-validation. And you can see is for intensity, you actually do, at least with the random forest, incredibly poorly until you have really several thousand features, or at least maybe several hundred. You don't do well at all with, say, 15 or 20 or 25. Had it in something like linear regression, this may have been different. You might have been able to just capitalize on a few of the most important features like molecular weight. But since random forest is each node, you only really get one decision. Is it lower than this value or is it higher than this value? It's really hard with random forest to exploit a feature that varies almost continuously along its whole domain. And but whereas in contrast with something like pleasantness, okay, maybe get a little bump when you're getting to using 10,000 features, but it's almost pointless. You can do almost just as well with like 10 features. And then some of the ones that maybe are more surprising, chemical, you get this big bump once you get above 1,000 features, or acid. Maybe some of the things that you maybe associate with some negative valence or with being very unpleasant. Most of the other features, you don't need thousands of most of the descriptors, you don't need thousands of features to predict accurately. You really probably only need a few dozen. But intensity is definitely very complex. In other words, there's probably many ways for something to be intense, but there's not very many ways for it to smell like wood. So another variant in terms of predicting, being able to predict the mean and the standard deviation is instead of doing median imputation and taking the mean, you can also mask the missing value. So instead of imputing that a missing value of for someone who couldn't smell something, and so instead of imputing the median for how woody it might have smelled had they been able to smell it, you just mask that and you take the mean of the remaining values across the other subjects who could smell it. So I'm calling that matrix Y mask, and it turns out that whether predicting mean or the standard deviation, one of Y or Y mask is the better summary to use. And why is that? Because there's some imputation bias in sigma. If I assume that someone who couldn't smell something smelled it a lot like the other people, I'm artificially deflating the variance. If I have values that are 25 and 75, that has a certain variance, but if I assume that someone who couldn't smell it smells it at 50 because that's in the middle, well, 25, 50, and 75, that has a smaller variance than just 25 and 75. So I'm screwing up the estimation of the variance by doing median imputation, whereas for the mean, it doesn't matter so much. So I'm going to use Y mask to compute standard deviations. I'm going to use Y to compute the means, and that's basically just the difference between imputation and masking for computing the average across subjects, or the variance across subjects. So the mean-variance relationship, if you plot the mean across subjects versus the standard deviation across subjects for a given descriptor, you get some interesting relationships. So what's it a name? That which we call a rose by any other name would smell as sweet. There's actually lots of different ways for something to smell sweet, but there's a very small number of ways for it to smell very, very, very sweet or not sweet at all. And so there's a very small number of ways for a mean to be almost the highest it could be, 100, or 0, where it doesn't have any value at all. So any perceptual descriptor that is a really high or really low mean must also have a really low variance. There's no way to get a mean of 0.0001 unless all the values are almost all the values are 0. And similarly with 100, almost all the values have to be nearly 100 to have a very, very high mean. So what does that look like? If you just plot the mean versus the variance of these descriptors, you get something like this parabolic shape. And this is for intensity, where you have quite a wide range of values from 0 to 100. Many of the other features you don't have quite as many large values, but you'll see the partial curve traced out. I'll show that in the next slide. So where have I seen a relationship like this before? I was a chemistry major, and so the next part of my career, I was a synaptic physiologist. And one of the techniques in synaptic physiology, as described in one of my favorite all-time textbooks, is something called non-stationary fluctuation analysis, which is basically just looking at the mean and variance relationships at different time points in the evolution of a synaptic current. And that looks something like this. So here's the downward going deflections there are excitatory postsynaptic currents, and these black lines sort of traced on top of them are the mean value across replications, and the upper trace in each of these 4 quadrants is the variance across replications. And so what does it tell you when you see the variance being of a certain level and the mean being of a certain level? And what does it tell you sort of before and after some experimental intervention? Well, it turns out that the mean and the variance have this similar kind of relationship, this parabolic relationship for synaptic currents, and that actually can tell you a lot about what's changed when you do some sort of intervention, like you try to potentiate the strength of the synapse, or you wash on some drug and it reduces the conductance of ion channels. And so different ways that this curve could evolve are indicative of different kinds of specific mechanistic changes underlying the strength of the synapse. And so I said, okay, well, let's just look up the equations that are used to describe this, and that probably just more or less describes what's happening, the mean, the variance in the perceptual data. And so if you can, they actually have a very specific theoretical relationship for this kind of data, where the mean is bounded, and it looks something like this. So the standard deviation is proportional to both the square root and the square of the mean. And so here are the data for all 21 different perceptual descriptors. And you see intensity traces out across the full range of the mean. The other one's less so, but nonetheless, if you simply fit to that equation, you get excellent fits. They're almost perfect, perfectly describe the relationship between the mean and the variance. And so using that, I realized that, okay, if I can predict the variance almost perfectly from the mean, then when I'm predicting the variance, I should probably use the mean. So since the mean contains information about it, it might be easier just to predict it directly. And part of what motivated this was when I was trying to predict the standard deviation of something like intensity data without using the mean, I was doing a pretty terrible job. And I think some of the other competitors experienced the same thing. It was actually pretty difficult. But if you simply use the mean and just pass it through this function, this parabolic function, you get a much better prediction, because the mean prediction of intensity is actually not so hard. You get correlations about 0.7 or higher. But standard deviation prediction directly it can be very difficult. So simply estimating the mean and then passing through this function to estimate the standard deviation turns out to work really well. How well that works compared to estimating the standard deviation directly depends on the descriptor. So I do cross-validation. I sort of estimated this parameter C, which is how much do I weight the direct estimation of the standard deviation from the descriptors and from the data versus my functional transformation of the mean estimate. And you get a different value of C for all the different descriptors. It's generally somewhat small, meaning you're mostly using the mean to estimate the standard deviation. I think this was helpful in getting the best performing predictions of the standard deviation. So why do we even care what the standard deviation is? Well, as Leslie and Andreas talked about earlier, you have this variability in how people rate something, and some of that is trivial. It's due to the mean. The mean in part determines the variance, but some of it's not. So all the residuals around that fitted blue curve are due to certain molecules, which each dot being one molecule, being over or under dispersed relative to the prediction you get from the mean. So in essence, a black dot, which is really below the line, is something that is way more homogeneous across people than you would expect, given its mean value. And something that's way above the line is something that's much more dispersed across people, much more variable across individuals than you would expect from that prediction. And so by subtracting that blue curve, which I don't have a slide for that, but if you subtract that blue curve, you get sort of, and get the residuals, that's sort of an estimate of variance that's not simply a function of the mean, but is really a function of human variation. So I also was wondering, especially after the challenge, and how well could someone have done on this challenge, right? Is there any way to estimate how well someone could have done? Well, we had a finite amount of data. I mean, it's a lot of data, but still 49 subjects is not a million subjects. And so if you want to try to predict how well someone could have done, you can simply do that by saying, how well do the subjects predict themselves? So you can do like training test splits just using half the subjects to predict the other half. So you already know how half the subjects rated that molecule. You just want to ask how the other half did. And it's not perfect because each subject has says variability both across replications, which you can see if you look at the replicates, but also just each subject is different, right? And so even with, say, 25 subjects in one cohort and 24 in another, you're still going to get some random variation, both in the training set and in the test set. And so it's impossible to predict perfectly, even with a perfect model with a godlike understanding of olfaction, which I certainly don't have. So what would that look like if you had that godlike understanding of olfaction with this data set? How well could you have done? And the blue line traced across the 21 features shows that essentially intensity it should be able to predict almost perfectly, even with a finite data set, which suggests we're still probably a lot to be gained there. And for the other features, you can see varying levels from about 0.5 to 0.8. So part of that comes from just what's the human variation across subjects for these different features. The red line is how well I did, and I think similar to how a lot of the other performers did as well, which you can see it actually sort of traces out a path that's similar to the blue line, it's just worse, which sort of tells you that the variation in performance across descriptors that we observed is not that some descriptors are just really hard to understand on a molecular basis, but is that there's just a lot of variability across subjects. If there's variability across subjects, there's just only so well you can do the prediction. If you had say, a million subjects, I think instead what you'd see is the blue line would be, I'm not sure where it would be. It would be, I guess you imagine it'd be at 1, and then the red line would probably be a lot flatter than it is now. So this is what it looked like for standard deviation. It's sort of similar, where it's related to the best possible prediction, but worse. And so this plot on the right-hand side, these purple dots, one for each feature. So it's the best possible prediction is on the x-axis, and then the prediction I obtained, the quality of the prediction I obtained is on the y-axis. So of course every dot is to the right of the dotted line because they're all worse than the best possible prediction, but they do sort of trace out a line which suggests that they're all similarly worse than the best possible prediction, which again just sort of is recapitulating what's in the first panel. I don't think any of the descriptors are in principle harder than any of the others with enough data. But with finite data, some of them are extremely challenging because of the variability across subjects. And here's again what that looks like for the standard deviation. So I think this has been a great advance in trying to predict smell from shape. I think this is as Yuanfeng showed in her slide, which I didn't go into, but I think Amit may go into it a little bit later. There are other physicochemical descriptors besides what's just in the Dragon data set. There's the name features that Yuanfeng used, and there's also some other descriptors that we just explored in the collaborative phase of the challenge, which further boost the performance of the models. Dragon 7 will be coming out, as Andreas mentioned, in a few months. Maybe it will have some more of those features. Maybe Dragon 8 will have even more. And maybe if we had a better understanding of chemistry or of the receptors, we'd have even better ones. I think if we had a challenge or a data set that contained something about the binding of all the molecules, say, in this challenge, or at least a set of molecules that spans the kinds of molecules that are odorous to all the functional human receptors, this would be, we have subsets of this data, in part from work by Hiro and Joel and Andreas, but we don't have the complete set, right? It's still impossible to predict for an arbitrary molecule how well it will bind to an arbitrary receptor. But the closer we get to that, and we can use that as an intermediate stage, I think it's going to be much more effective than trying to predict olfaction from the type 3 van der Waals, Svengali interaction that is in these Dragon descriptors, where I don't know it's really hard to understand what the relationship between that is and olfaction, but saying this binds to this receptor here and this is how well, even if that's a noisy prediction, is going to be incredibly helpful. And not only will that help us predict what things smell like, it will also tell us how much of the variability in human olfaction occurs at the level of receptors, at the level of the olfactory bulb, at the level of the piriform cortex, and deeper levels still. We don't really know the answer to that. We don't know how important some of these other structures are in determining why things smell the way they do, or how much of that is just done at the level of the receptor. But I think we're starting to get near the prediction limits. So you saw on the previous slide about how well we're doing. I think it's pretty good. I think people can debate that though. But I think we're ready to start putting our models to some scientific use to answer scientific questions that many of us in olfaction are interested in. So why does feature F predict descriptor D? Why does, molecular weight is an easy one. Things that are low molecular weight are the only things that are volatile, so we can smell them. But why do some of these other descriptors have anything to do with, woodiness or valence? Sometimes it's hard to tell. Even someone with a knowledge of chemistry and a knowledge of receptors might not be able to figure it out, but at least we know where to look now, because from these models we can identify the specific features, and it's not very many, that were involved in say, the prediction of woodiness or grassiness. And what's the dimensionality of smell perception? So the better we can predict the molecules, I think the better we can ask these questions about how high or low dimensional smell perception really is, because it's a question we still don't know the answer to. So I'd like to acknowledge all the people who made this possible. So Andreas and Leslie and all the human subjects, and now Peggy I learned about is another person who's helped collect the data. This is a really fantastic data set. It's one of the largest, if not the largest, data set in the history of human olfaction. And Andreas mentioned there was a data set from the Atlas of Human Olfaction before this, which is just much smaller. So I'm really indebted to them for their creation of this data set. And let's see here. Pablo and the other administrators and developers. Pablo answered many incessant questions that I had, and anyone who's active on the forums as well. All the other collaborators and participants who, especially in the community phase, I think we learned even more about what kind of descriptors would be useful and what kinds of techniques would be useful, including, and I wanted to just comment on one of Yuan Feng's points about linear models versus these decision trees. It turns out that the people who did take the linear models approach do not that much worse on most of the descriptors, and actually somewhat better on predicting intensity, which it was surprising to me, because I thought this should be a really nonlinear kind of problem. And I think when you just goes to show, when you add a lot of different things together, sometimes you get something that turns out to be linear in the end. But it still remains to be seen as we add more descriptors and more. Whether this will turn out to be really seen as a linear or nonlinear problem. All right, I hope I have the other acknowledgements memorized. Who else did I want to acknowledge? There was someone else at the bottom. I'm sorry if I forgot who you were. One of them was all the contributors to open source software that made this possible. I think software developers don't get enough credit in the scientific chain of scientific development. So that's all. Thank you.
Any question? Yeah.
So you mentioned that one of the problems you thought was that there weren't enough subjects. So if you had 1 million subjects, you'd do much better.
Oh, you would definitely do better, yeah.
Right. So the question is, do you do anything analytical there where you used a subset of the data to see how well you did with 5 subjects versus all of them?
I didn't do that. Essentially, I just presented those curves. Yeah, here was that last thing. All I did was was this. So essentially, if I had an infinite number of subjects, I believe that blue line would be at the top. It would be all at 1. Because then there's no finite data limitation to how well you can predict. I don't know that you can in practice actually predict that well. Probably not. But essentially, what's above the blue line, it's a limitation due to subjects, and what's between the blue and the red line is a limitation due to imperfect understanding of olfaction and imperfect predictors and algorithms and people. So.
And I guess at what point do you reach diminishing returns, right? So 100 subjects, 500 subjects.
That's a good question. I didn't actually look into how how that blue line moves up and down quantitatively with a number of subjects. I'm not necessarily calling for another 49 or 4,000. I'm just pointing out that putting the degree of prediction that we had in context of versus how well we could have done.